One of the most often cited arguments against GMOs is that they are not natural. The scientific community has been working to poke holes in this argument by looking to history. A May 2015 study published in the Proceedings of the National Academies of Sciences was just the ammunition GMO advocates needed to demonstrate that nature, and more specifically bacteria, have been modifying genes for thousands of years. The authors of one paper found that transferred DNA sequences were identified in sweet potatoes, showing that an agrobacterium infection occurred in evolutionary times. Agrobacterium transfers DNA fragments, and plant biologists have since adopted this natural mechanism to use as a vector for developing GM crops. The paper's authors believe that their findings that the sweet potato is a naturally transgenic and traditionally consumed food crop could positively affect the current consumer distrust of the safety of transgenic food crops. They write that this remarkable observation may change how we govern the unnatural status of transgenic crops. But does this argument stand up to scrutiny? Maybe not. Agriculture in general isn't natural. Human civilization has survived and grown because of our ability to domesticate crops and livestock to feed ourselves. That alone should raise questions about how we use the term. Many GMO supporters suggest that we've been selective breeding and crossbreeding for thousands of years, and that therefore genetic modification is not new, and current debates are a waste of time, and they stand in their way for helping to feed the hungry with their technology. This argument is condescending and counterproductive. It's condescending because it implies that not only do people worried by laboratory manipulation of genes have nothing to fear, but that they are unintelligent and wrong. It's counterproductive because it's inefficient, polarizing science communication. There are many ways to genetically modify plants. These range from simple bacterium vectors already described, to using a gene gun to shoot DNA-coated gold particles into plant cells, to CRISPR-Cas9 technology, a new molecular editing system that uses a scissor-like enzyme to alter particular DNA sequences within a single genome. This is very different than traditional plant breeding that you do in a high school biology lab, and this difference should be taken seriously. Scientists look to this technology to improve nature. GM and gene editing may by nature be more precise, which is a major benefit of the technology, but that also inherently makes it different. As journalist Tamar Haspel argues, the constant defense of GMOs as natural undermines one of the good arguments advocates actually make, that the tools and techniques used to insert and move around individual genes has enabled scientists to make changes in organisms that are much more predictable than the changes that come from arbitrarily crossbreeding two organisms. Haspel says that this argument works only if you admit from the get-go that transgenic breeding is materially different from what we've been doing for thousands of years. And here's the basic paradox. The minute we introduce patent and intellectual property of life forms, we do admit that these modifications are different. Perhaps our conceptions of nature and of genetic modification are too simple.